Good morning, everybody. Let's get started. Today, what we're going to do is to talk about ketone genesis, ketone body synthesis. Probably we'll also have time to talk about the cholesterol synthesis, right? And we started this slide last class, as I remember, right? So synthesis of a ketone body. So when are we, our body started to synthesize ketone bodies? We know during the fasting, right? And we also mentioned not just a short period of time of, duration of starvation, fasting, but usually when ketone genesis becomes very active, usually during the long-term starvation, for example, we starve for longer than 2 days, and then our body, start synthesize lots of ketone bodies. And that was a physiological condition. And also ketone genesis can occur under the pathological condition when we have a very high levels of fatty acids in the circulation. For example, we mentioned, right, type 2 diabetes or diabetic conditions. And in which place, which tissue organs that synthesize ketone bodies? You remember? Liver, right? Liver. Everybody knows the liver. And also, you wanted to know a little bit more detailed, specifically in the mitochondria, right? So in the liver cells and mitochondria, in the mitochondria, where the ketone bodies are synthesized. And also talk about this pathway. Fatty acids, right? During the fasting condition, we have a lot of fatty acids available. Basically, it's mobilized from fat tissue, right? And through the lipolysis activities. So, and once we have lots of fatty acids available in the circulation, which tissues pick up most of fatty acids? Remember? Which tissues? Muscle, yeah, skeletal muscle, right? And also heart, cardio muscle. So pick up lots of a majority of the fatty acids, right? Two-thirds, like about 70% of the circulating fatty acids are picked up by muscle, and undergo fatty acids oxidation, or also we call it beta oxidation, right? So what is the final product of a fatty acid oxidation, beta oxidation? Beta oxidation. Acetyl-CoA, right? Acetyl-CoA is the final product of a beta oxidation of fatty acids. And also thinking about one important steps for the beta oxidation. When fatty acids are ready for oxidation, the first step is to activate to fatty acyl-CoA, right? In the cytosol. Where does beta oxidation occur? In mitochondria, right? So fatty acyl-CoA needs to be moved from cytosol into the mitochondria, which is a key enzyme that helps transport the fatty acyl-CoA from cytosol into the mitochondria. Remember? Transporter, system, right? I just ask you, we have 4 enzymes, right, located on the in the mitochondrial membrane, in our mitochondrial membrane. I just ask you to remember one key enzyme that, CPT1, that helps transport fatty acids from cytosol into the mitochondria, right? So and for the beta oxidation. So once fatty acids been oxidized to acetyl-CoA, in the muscle, acetyl-CoA will enter the TC cycle, right, for complete oxidation, for ATP production. But in the liver, the acetyl-CoA will not enter the TC cycle for oxidation, complete oxidation, but to ketogenesis pathway, right? So again, the acetyl-CoA is the substrate of a ketone body synthesis. So ketone body is also synthesized from acetyl-CoA molecules, right, in the liver. So ketone body then is released into the circulation and picked up by other tissues for use as energy. So which tissues you think is the most important tissue that utilize ketone body as an alternative fuel during fasting? Brain, right? Brain and also muscle, right? Now let's talk about ketone body. So the molecules, ketone body molecules. So our body actually have a very high levels of beta hydroxybutyrate and acetoacetate in the circulation, right? So basically, these are the two main ketone bodies, right? Again, acetoacetate and beta hydroxybutyrate are the main ketone bodies, and acetone basically is the byproduct, right? So acetone can be synthesized or converted from acetate. So acetate can spontaneously convert to acetone. So acetone basically is excreted in the lung, right, immediately. So not have much acetones remain in the body, right? And also that makes a kind of special smell if you know that particular type of 2 diabetic patient, under the ketoacidosis, right? You smell like fruits or kind of special nail polish, this kind of smell, right? So this is because of acetone, it's excreted in the lung. And the acetoacetate and beta hydroxybutyrate, this can also interconverted, right? But again, these are the two main ketone bodies that have a high levels in the body. And the synthesis pathway, again, the starting molecules of ketogenesis is the acetyl-CoA, right? So acetyl-CoA we know is basically is from the beta oxidation of fatty acids, right? And as one molecule of acetyl-CoA can together with a acetyl-CoA, which is a 4-carbon molecule, is the second product of beta oxidation, right? But it can also be formed from 2 molecules of acetyl-CoA. So this acetyl-CoA together with acetyl-CoA forms a molecule called hydroxyl 3-methylglutamate-CoA, or just simply called HMG-CoA, right? So HMG-CoA is synthesized from 1 molecule of acetyl-CoA and 1 molecule of acetyl-CoA. And the enzyme catalyzes formation of HMG-CoA is HMG-CoA synthase, right? So this is the reaction. Actually, we also will talk about later in the cholesterol synthesis. So the common pathways, right, or reactions for both cholesterol synthesis and ketogenesis. And once HMG-CoA is synthesized, that it can be broken down by an enzyme called HMG-CoA lyase, and to release the acetyl-CoA that can be recycled, right, for the next round of HMG-CoA synthesis. But it also release a ketone bodies, acetic acetate, and this can be converted to hydroxyburate, right? Both molecules can be interconverted. So again, here what you need to know, this slide is first the substrate or starting molecules, right, of a ketogenesis is acetyl-CoA. And this acetyl-CoA basically is mainly from fatty acids oxidation, right? And secondly, the common pathways for both ketogenesis and cholesterol synthesis is the formation of HMG-CoA from acetyl-CoA and acetyl acid-CoA, right? Okay, I already tell you now, but so liver we know is the main tissue that synthesizes ketone bodies, right? But liver cannot use ketone bodies. I'm going to tell you the reason why later. So once a ketone body is synthesized, ketone body just can simply diffuse, right? Release into circulation, out of the get out of the liver and diffuse into the peripheral tissue like brain and muscle and other tissue, right, for use as energy. And this gives you the overview of ketone body synthesis and utilization. So liver is the main site for ketogenesis. So ketone body is synthesized from acetyl-CoA, and fatty acids oxidation is the main sources of acetyl-CoA, right, for ketogenesis. But glucose can also contribute a little bit of acetyl-CoA, but it's not the major sources, right? Thinking about ketone body, ketogenesis usually occurs during the fasting, right? So we were not going to oxidize a lot of glucose, so that's, but mainly fatty acids. So once it's synthesized, a ketone body from acetyl-CoA, then ketone body released into the circulation of blood, and some of the ketone bodies can also excrete in the urine, but some acetone, as I mentioned, right, excreted in the lung. So the other main ketone bodies can be picked up by peripheral tissue used for energy. So in the peripheral tissue, particularly in the brain and skeletal muscle, ketone bodies can be used simply just by the pathway that break down the ketone body again to the acetyl-CoA, and then acetyl-CoA enter the TC cycle for energy production, right? Let's just simplify the diagram, show you how ketone bodies synthesized in the liver and transported into the circulation and picked up by other tissues for use as energy. And this here I'm going to talk about the utilization of ketone bodies. Take the muscle as an example. Of course, your brain is also the main tissue that utilize ketone bodies, right? So ketone bodies is two main ketone body molecules. The very important enzyme that break down the ketone bodies to acetyl-CoA and acetyl-CoA is the enzyme called acetyl-CoA transferase, right? So succinyl-CoA transferase can break down acetoacetate to acetoacetal. Sorry, this is not to the transfer the CoA group to the acetoacetate and make it become the acetoacetal CoA, right? And then the silase is an enzyme that break down the acetoacetal CoA to acetyl-CoA, right? But succinyl-CoA transferase is the key enzyme for ketone, for utilization of ketone bodies, right? And acetyl-CoA eventually entered TC cycle, right, for ATP production. So this is a key enzyme, and it's also utilized the ATPs, right, for this reaction. So succinyl-CoA transferase, again, is a key enzyme for utilization of ketone bodies. And the liver, I mentioned earlier, makes ketone bodies but cannot utilize ketone bodies. So the reason that liver cannot utilize ketone bodies is because, you guys probably already know, right? Because what? Yes, lack of this key enzyme. So liver does not have a succinyl-CoA transferase, so liver cannot break down the ketone bodies and utilize ketone bodies, right? Okay, questions? Any questions? No? All right. So now let's look at the energy production from palmitate. So let's say in the muscle, we don't have a problem, right, to pick up fatty acids and oxidize fatty acids and complete oxidize, right, through TC cycle for ATP production. And we already calculated, right, for oxidizing one molecule of palmitate, will generate 8 acetyl-CoA molecules, which will enter TC cycle and for complete oxidation and produce CO2 water. And we know that can generate 129 ATPs, right? If you still remember. So, and what if in the brains, you know, brain cannot utilize fatty acid, right? Ketone bodies will be the molecule that brain utilizes for ATPs. So see how many ATPs can be generated from start from oxidation of palmitate and generation of ketone bodies and utilization of ketone bodies. So for all this process, eventually will generate only 125 ATPs, right? So we have less ATPs be made if fatty acids oxidized goes through the ketogenesis pathway, right? Because of the utilization, if you remember, process utilize ATPs, right? So you have to minus the 4 ATPs being used for breakdown ketone bodies and for oxidation. So so again, the ketogenesis and utilization of ketone body pathways produce less ATPs. But our body does this again because of brain cannot uptake the fatty acids directly, right? So we have to, go sort of indirect, making ketone body first and then utilize in the brain. Questions? No? Okay, now let's talk about regulation of ketogenesis, right? So we only want a ketogenesis occur when we need it, right? We don't want it to, do something unnecessary. So there are actually 3 key points that regulate ketogenesis. First is the lipolytic pathway or lipolysis rate, right? Which makes sense when during the fasting, right? When we need a ketogenesis, so when we need to break down first the triglyceride, right? Mobilize triglyceride, release fatty acids. So lipolysis and fatty acids levels in the circulation increase. This is one important driving force for ketogenesis, right? Again, lipolysis rate increase and free fatty acids level in the circulation increase. That is one important factor driving ketogenesis, right? And a second important point is the CPT1 gateway. We know CPT1 is a key enzyme, right, that control transport of fatty acyl-CoA from cytosol into mitochondria for beta oxidation. So when we need higher levels of ketogenesis, then we wanted to push more fatty acids, right, get into the mitochondria for beta oxidation. So the CPT1 is the second key point that regulates the ketogenesis, right? When CPT1 levels increase, then we have more fatty acids being transported and increase the beta oxidation and increase the ketogenesis, right? And the last point is the acetyl-CoA levels. We know the ketone body is made from acetyl-CoA molecules, right? So when we have high levels of acetyl-CoA, or we have more acetyl-CoA available for ketogenesis, so acetyl-CoA levels is also is important regulating point, right? They can regulate ketogenesis. But these 3 things all kind of connect together, right? When we have increased lipogenesis, we have increased CPT1 activity, we have more fatty acids being available for beta oxidation, then generating more acetyl-CoA for ketogenesis, right? So just try to remember this 3 key regulatory points. Okay, so ketogenesis, relatively simple, right? The pathway, you just need to know when does this occur and where does this occur, and then how this which molecules are the substrate for ketogenesis, and how ketogenesis activities is regulated, right? And which tissues utilize ketone bodies, and why liver cannot utilize ketone bodies, right? Something like this. Questions? Okay. Now let's talk about the ketoacidosis. This something is not a physiological, right? It's kind of pathological. We have a certain levels of ketogenesis during the prolonged fasting, which is good, right? Which is called adaptation, a physiological adaptation of our body, try to make the alternative energies for tissue use. But under the pathological condition, the ketoacidosis is considered a pathological metabolic state, right? And that is not normal. So ketoacidosis usually had a very or extremely uncontrolled ketogenesis. That you have a very high ketogenesis that is definitely exceed the physiological levels, right? The ketogenesis, and generate very high concentrations of ketone bodies. And ketoacidosis is mostly common in the uncontrolled diabetic conditions, and also prolonged alcoholism. So there's two specific conditions that is very common to have a ketoacidosis. So here I just wanted to give you a little bit detailed, sort of mechanism why under the uncontrolled diabetic condition or alcoholic condition, the ketoacidosis occurs, right? Happen. So under the both diabetic and alcoholism, causing the problem, that means the cells in the body cannot utilize glucose, or glucose utilization somehow has a problem under the diabetic or alcoholic condition, right? And these two different conditions have a little bit different mechanism. For example, under the type 2 diabetic conditions, we know, if you know, I'm not sure if you guys know what is the main cause of the type 2 diabetes. Mechanistically cause. Do you guys understand? And type 1 is because of beta cell cannot secrete enough insulin, right? And what about type 2 diabetic patient? Diabetes. Do you know what is the cause? No? Okay. So insulin resistance. Have you heard about insulin resistance? Okay. So type 2 diabetic patients at insulin secretion, particularly at the early stage, they are okay, right? They be able to secrete insulin at insulin level even higher than the normal people, but the insulin action actually reduced. That means the insulin cannot see its function efficiently, right? So so-called insulin resistance. So insulin, we know the main function of insulin in terms of the glucose metabolism, glucose transporter, if you should remember, which, GLUT4. GLUT4 is the insulin-dependent glucose transporter, right? So when insulin resistant, that means the GLUT4 cannot be efficiently translocated from inside of cell to the cell surface. So glucose transport or uptake is decreased under the diabetic conditions, right? So fat cell or muscle cell not able to uptake a lot of glucose from circulation into the cell for energy use. So your loss of glucose remain in the circulation, causing the high blood glucose. But inside of cells, glucose level actually relatively low because can transport glucose into the cell for energy. So inside of cell, glucose levels are decreased. So causing kind of energy crisis, right? Inside of cell. So that makes body to break down more triglyceride, release more fatty acids, right? For alternative energies. So that in diabetic patients, usually have high levels of fatty acids in the circulation. So this is one of the key regulatory point, right? Regular ketogenesis, we just talked about it a couple slides early. So in that type 2 diabetic patient, you have, because of the high levels of fatty acids, that can increase the ketogenesis, right? And for the alcoholism, for kind of the problems that the gluconeogenesis has a problem. So the alcoholic conditions, gluconeogenesis cannot be efficiently, glucose cannot be efficiently synthesized through the gluconeogenesis. So also causing the low blood glucose level, right? So this will sort of mimic the fasting condition. So then also push a lot of fatty acids being mobilized from fat tissue, right? Causing, fatty acid levels increase, and similarly, causing the ketogenesis increase and produce a lot of ketone bodies, right? Does that make sense? And this is a slide and show the alcoholism, how alcoholism blocks or decrease the gluconeogenesis. So ethanol, and this is the metabolic pathway of ethanol. Ethanol can be metabolized to acetaldehyde and acetate, and to generate a lot of acetyl-CoA. And the two reactions in this pathway requires NAD, right? And NAD can, during the ethanol metabolism, NAD is needed, and because during the alcoholism, and this pathway being increased, right? So we have a increase the need of NAD. So NAD basically is produced through these two reactions, right, or metabolic pathway. One is the conversion of pyruvate to lactate, if you still remember, right, and can utilize NADH and generate NAD. And the other pathway is that oxaloacetate being converted maline, and this reaction also utilize NADH and generate NAD. So this NAD basically, provided for ethanol metabolism. So because of this reaction is increased, right, you need more NAD. So the oxaloacetate will be utilized a lot for generating NAD, so that causing the decrease of oxaloacetate. So oxaloacetate is an important substrate, right? Remember, for gluconeogenesis. So once you have a decrease the oxaloacetate, and gluconeogenesis, it cannot, be efficiently synthesized, right, to glucose. Synthesize glucose. Okay, any questions? Okay, now let's talk about the regulation of a long-chain fatty acids oxidation. So I'm going to divide it at the fasting condition, right, and see how fatty acids oxidation is regulated, because this pathway is also important for for acetyl-CoA production, energy production, and also for ketogenesis. Let's look at the fat condition first. Under the fat condition, right, we have high levels of glucose. That's no problem with the energy that from glucose. And also because of a high blood glucose level, I also increase the insulin. If you still remember this reaction, the lipogenesis reaction, right? The key reaction, the lipogenesis. The first key reaction is the conversion of acetyl-CoA to malonyl-CoA, and this is a key enzyme, ACC, right? So ACC is stimulated by insulin when insulin level is high, and once the ACC activity is increased by insulin, and will causing the increased lipogenesis, right? So you will expect or have a very high levels of malonyl-CoA be generated. So, and try to remember malonyl-CoA is the very important substrate that can regulate the fatty acids oxidation, particularly regulates the transport of fatty acids, right? So CPT1 is the key enzyme that are located on the mitochondrial membrane, right? This represents the mitochondria, this is the cytosol. So malonyl-CoA is a negative regulator of CPT1. So mean that when we have a high malonyl-CoA levels, the CPT1 activities goes down, right? And so once CPT1 levels goes down or activity goes down, then what happens to the fatty acids oxidation? What do you expect? What do you expect? Fatty acids oxidation goes down, right? So you have a less fatty acid or fatty acyl CoA being transported into mitochondria, so you have less acetyl-CoA available for beta oxidation. So fatty acids oxidation will be downregulated, right? So that also makes sense, right? During the fasting condition, when we have enough glucose for energy, then we don't need to break down or oxidize fatty acids for energy. So this sort of the regulated pathway, you know, how the fatty acid the fatty acid oxidation being inhibited during the fasting, during the fat condition, right? And started with increased glucose, insulin levels, and this is a key thing, you know. Malonyl-CoA is a key molecules, right, being upregulated or increased in the fat condition, and then this will be the inhibitors of CPT1. Right? Any questions? Okay. Shall we take a break? Okay. Okay. Now, let's see what about under the fasting condition, how fatty acids oxidation is regulated. Oh, sorry, I haven't completed all the stuff. So, all right, that was for fat condition, right? Highlighted in red. And then let's look at the fasting condition. During the fasting, because we have a lower blood glucose level, right, that will stimulate the fatty acids mobilization. So we have high levels of fatty acids due to the high rate of lipolysis, right, fat mobilization or triglyceride mobilization. And that will cause the increased acyl-CoA levels in the circulation. When we talk about the high-fat diet, inhibits lipogenesis. If you still remember, acyl-CoA level, right, in the circulation is also important regulators of a key lipogenesis enzyme, ACC. You still remember this, right? So how does long-chain acyl-CoA regulates ACC? Inhibits or increase? Inhibits, right? So inhibits ACC. So inhibits lipogenesis, and because of ACC activities inhibited, so what do you expect at mineral-CoA levels under the fasting condition in the cell? Will be decreased, right? Because of ACC activities inhibited. So what about CPT1 activity? If mineral-CoA levels decrease, up, down. Again, I'll tell you, mineral-CoA is negative regulators of a CPT1. When you have high levels and CPT1 activity goes down, when you have low levels of mineral-CoA and CPT1 activity goes up, right? So negative regulator mean, you regulate opposite way. So when we have lower mineral-CoA levels under the fasting condition because of ACC activity being inhibited by fatty acyl-CoA, right? So we will expect CPT1 activities increase, which will make more fatty acyl-CoA being transported into mitochondria, right? So we will have higher levels of beta oxidation, so that will generate more acetyl-CoA levels, right? So all this together definitely will promote ketogenesis, right? Does that make sense? So just try again, remember the key things here, the regulation, right, under fat fasting condition. One is this reaction, the key reaction in lipogenesis. The ACC catalyzes the formation of mineral-CoA, right? So, and how ACC is regulated by hormones, insulin, and also the fatty acyl-CoA levels. And the second key thing is the mineral-CoA. So mineral-CoA level is very important in negative regulators again of CPT1, right? So once you remember these two key things, then you will be easy to understand how the fatty acid oxidation being regulated, right, under fat fasting condition, right? But in the big picture, you can just easily thinking, under the fat condition, we have enough glucose. We don't need to utilize fatty acids, right? So you will expect fatty acid oxidation activity will be decreased. Ketogenesis will also not active, right? But fasting condition, we need to utilize fatty acids for energy, so we will expect high activities of fatty acid oxidation and high levels or activity of ketogenesis, right? And once you have this big picture, and then you can go to the little bit detail how, this activity is being regulated by specific hormones and substrate, and pathway and stuff. Okay, so I think I'll pretty much finish the ketogenesis, fatty acid oxidation, ketogenesis, right? So this is something that you guys need to know, right? Ketone bodies basically is made in the liver from acetyl-CoA, right? The acetyl-CoA mainly is from the fatty acid oxidation, right? And ketogenesis, ketone bodies are synthesized in the liver, but liver cannot utilize because of lack of liver lacks the succinyl-CoA transferase, right? Key enzyme for ketone body utilization. And the brain and muscle will utilize most of the ketone bodies. And regulation of the ketogenesis, I mentioned the 3 key points. Can you repeat the 3 key points that regulate ketogenesis? First, and anybody? Fatty acids levels, right? Lipolysis rate and fatty acid levels. The second key point. Yes, numerically regulates. Yes, regulates CPT1, right? Activity. And last, acetyl-CoA levels, right? So how about if we have high levels of fatty acids, what do you expect the ketogenesis levels or activities? If we have high levels of fatty acids, goes up, right? Stimulate ketogenesis, and similar for CPT1 activities and acetyl-CoA levels. If they are all high, then we will expect a high activity of ketogenesis. Okay. Okay, so next we going to talk about the cholesterol synthesis and lipoproteins, but I think we can just finish cholesterol synthesis. We'll leave the lipoprotein next time, right? So cholesterol synthesis. So in our body, cholesterol present in 2 forms: free cholesterol and cholesterol ester, ester 5 form, right? So cholesterol ester, if you still remember, I talked about it, right? Basically, it's free cholesterol together with fatty acids. So one fatty acid molecule is attached to the cholesterol molecules formed ester, cholesterol esters, right? And present in most tissues and also plasma. And cholesterol has a good and bad side. And good side of cholesterol is essential structure components of a membranes, like cell membrane or organelle membranes. Yes. Oh, I'm sorry. Okay, sorry. Again, right? Cholesterol present in the 2 forms: free cholesterol or cholesterol esters in tissues and also in the plasma, right? And cholesterol has both good and bad side. So good side, cholesterol, we need cholesterol because cholesterol is essential, right? Structure component of the membrane, cell membrane, and also organelle membrane, like mitochondrial membrane or ER membrane, whatever. So and also cholesterol is the important the component of the outer layer, the lipoprotein that we're going to talk about later, right? So lipoprotein, and the other important side or good side of cholesterol is the cholesterol is a precursor of or other steroid hormones, for example, corticoid, steroid hormones, sex hormones, and vitamin D, bile acid. These molecules are all synthesized from cholesterol, right? So cholesterol has a good side that we need cholesterol, and bad side. Bad side that cholesterol is, you guys know, is a major constituent of a gallbladder, the stone, right? So, and the other bad side is that the cholesterol actually involved in the atherosclerosis, right? So if we need a cholesterol, just right amount, we don't need extra cholesterol. So if we have extra cholesterol that cannot be dealt with or cannot be disposed, then we're causing some health issues, right? Atherosclerosis or gauze, stones or something like this, right? So about the sources of cholesterol. So in our body, half of cholesterol in our body from synthesis, the de novo synthesis. That means our body can synthesize a little bit more than 50% of body cholesterol, right? So again, a little bit more than 50% of our body cholesterol is synthesized by our own bodies, and a little bit less than 50% of our body cholesterol from diet, right? So you can see the contribution. Diet contribute to our body cholesterol a little bit less than 50%, right? And biosynthesis. So 10% of the total synthesis cholesterol occur in the liver and intestine, right? 10% occur in the liver and 10% occur in the intestine, right? And all the cells that has a nucleus are able to synthesize cholesterol, right? And cholesterol usually synthesized in the ER and cytosol, right? Again, cholesterol is also synthesized from acetyl-CoA. So acetyl-CoA is an important molecule, right? It's a substrate for, can you tell me? I can tell you 3 lipids that synthesize from acetyl-CoA. 1? One is fatty acids, right? Lipogenesis we learned. And second, we just finished ketone bodies, right? And third, we just we're now talking about it, cholesterol, right? So acetyl-CoA is a very important molecule or substrate for fatty acid synthesis, lipogenesis, right? For ketone body synthesis and cholesterol synthesis. And transport. So cholesterol transport is very important when we have synthesized a little bit too much, too much cholesterol. The cholesterol need to be disposed, right? To transport from one tissue to the other tissue to dispose. So the liver is the main tissue that disposes the cholesterol, right? And cholesterol transport requires lipoprotein. We're going to talk about it soon. We have a different type of lipoprotein in our body, and LDL, the low density lipoprotein, right, is the lipoprotein that transport cholesterol into the cell, right? And HDL, the high density lipoprotein, the lipoprotein that transport cholesterol from peripheral tissue to the liver for disposal, right? So these 2 type of lipoprotein basically deal with the cholesterol homeostasis, right? And we didn't talk about chylomicron and VLDL because these 2 lipoprotein basically transport primarily triglyceride, right? So we have different type of lipoprotein, and each lipoprotein has their own function, right? And a different function. So cholesterol synthesis, you don't need to remember all the steps, but some key things, right? You need to remember. We can just go to the step by step, right? So first step is the synthesis of mevronate from 3 molecules of acetyl-CoA, and the key enzyme for this step is HMG-CoA reductase. So, HMG-CoA reductase is the key enzyme for whole cholesterol synthesis pathway, right? And I mentioned earlier, this is, if you probably looks like familiar, right? We just talked, you know, on the ketogenesis. So this is the same reactions as the ketogenesis. So again, this is usually the common reactions for both ketogenesis and cholesterol. And so once the HMG-CoA is synthesized from these common reactions, right, and HMG-CoA being converted to mevronate, and this is the enzyme that carries out this conversion, HMG-CoA reductase. So HMG-CoA reductase, again, is a key enzyme for cholesterol synthesis. And you also probably wanted to know how this key enzyme is regulated, right, by several things, like first the bile acid, cholesterol levels itself, and also mevronate. This is a substrate, right? One of the intermediate, metabolites that all these 3 things can inhibit reductase, HMG-CoA reductase, so then can block or inhibit the cholesterol synthesis, which makes sense, right? For example, we already have a high level of cholesterol. We don't want to synthesize more. So if we have a high levels of cholesterol, that will causing the inhibition, right? The inhibition of the key enzyme, and then inhibits cholesterol synthesis. And the other thing is that the drug, the statin is the cholesterol lowering drug, right? Basically, to lower cholesterol level. And the mechanism for statin is it's the inhibitors of HMG-CoA reductase. So basically, statins inhibit this key enzyme and to reduce the synthesis of cholesterol in the body. And this is the second step, the formation of isopentanediol diphosphate, and this involves the 3 sequential phosphorylation and 1 decarboxylation reactions, and utilize 3 ATPs, and these are the key kinase, right? Catalyze the 3 sequential phosphorylation reactions, and this is the phosphomevronate decarboxylase, right? Catalyze decarboxylation. But you don't have to remember all this, right? Just walk through these steps, right? And the next step 3 is the sequoline is formed from isopentanediol diphosphate, right? And involves the several reaction, isomerization, condensation, and reduction, right? And it requires the NADPH. Step 4 is lanosterol is formed from sequoline, and these are the 2 enzymes catalyze these steps. And this is the last step, right? The formation of cholesterol from lanosterol, and the isomerase and reductase, right? Catalyze this formation, the formation of cholesterol is the final step. So this is the overall, the reactions for the cholesterol synthesis starting from acetyl-CoA. Involves these 7 enzymes and utilizes 8 NADPH and 3 ATPs. And again, this is a key enzyme for cholesterol synthesis, HMG-CoA reductase, right? And homeostasis. So cholesterol, we know that a little bit more than half, right, synthesized by our body, and a little bit less than 50% of our cholesterol from diet. So the cholesterol level in our body need to be maintained at the normal range, right, a normal level. So this involves sort of the balance, between the cholesterol synthesis and the disposal. So synthesis of cholesterol, the regulation, so both processes need to be tightly regulated, right? When we need some cholesterol, we need to synthesize. When we don't need cholesterol, we need to dispose, right? So these two processes need to be balanced in order to keep our cholesterol level at a normal range. And for the cholesterol disposal, I mentioned a little bit, right? The LDL and HDL that involve the regulation of cholesterol disposal. And in terms of synthesis, and we just finished the synthesis synthetic pathway, right? And HMG-CoA reductase is a key that can control the synthesis of cholesterol. So when we wanted to control the cholesterol synthesis, we need to do something to regulate this key enzyme, right? So this are sort of all things that can regulate this key enzyme. For example, insulin and thyroid hormones, and these two hormones basically stimulate cholesterol synthesis by increase the HMG-CoA reductase. And under the other conditions like starvation, glucocorticoids, hormones, and glucagon, and basically inhibits cholesterol synthesis, right? Cholesterol, HMG-CoA reductase. And also these are the two important substrates. When these two substrate levels increase, can also inhibit the HMG-CoA reductase. And statin is the drug, right? Is also, HMG-CoA reductase is a target of the statin. It inhibits this enzyme. So this is the sort of overview of how cholesterol synthesis can be regulated by many things, right? So this is one last slide for the cholesterol synthesis unit. I just wanted to get a little bit in detail about the transcription regulation of HMG-CoA reductase. I'm not sure if you are familiar with this kind of diagram. What that means is, this is DNA sequencing, right? The HMG-CoA reductase, and the front of untranslation sequence of the HMG-CoA reductase. There is the DNA sequence called SRE, the sterol regulatory element DNA sequence, and this element can be bound by a transcription factor called SREBP, which is a sterol regulatory element binding protein, right? So this is a transcription factor that can control the transcription or expression of HMG-CoA reductase, right? And this here I just wanted to tell you how cholesterol or mevalonate regulates HMG-CoA reductase through regulating SREBP transcription factor activity or action, right? So for example, when we have high levels of cholesterol or mevalonate levels, SREBP transcription factor activity is being inhibited, right? Because of SREBP transcription factor is needed for transcription of a HMG-CoA reductase. So when we have a lower SREBP, then we have a lower activity here, right? So we basically cannot efficiently promote the transcription of this enzyme. Does that make sense? Right? So again, just try to remember the cholesterol or mevalonate levels increase, then inhibit SREBP activity and decrease transcription of HMG-CoA reductase, right? I think that's all for cholesterol metabolism, right? Pretty simple. All right, have a quiz.